Some researchers work on timescales of a picosecond, a millionth of a second, but not all of them. Take this ammonite, for instance. It is 100 million years old. Paleontologists also use synchrotron light to reveal the secrets hidden within the fossils.
Most paleontologists want to know what is inside their fossils without having to break them. It's understandable when the fossils are rare or very important or both. Thanks to the very special properties of the synchrotron X-ray beams, it's possible to see many anatomical details inside fossils without having to damage them. This piece of amber, for example, is 100 million years old and is completely opaque. There's no way of knowing if there is a fossil inside or not. Using a synchrotron beam, we can see if there is something inside, and we are also able to scan it. Here you have a fossilized wasp. The original size is less than 3 millimeters. We can study in detail the anatomy of these fossils, which disappeared 100 million years ago. This is one of the largest dinosaur eggs that exist, and there you have one of the smallest. Thanks to the synchrotron X-rays, we are able to look inside the preserved embryo's skeleton and are able to virtually separate all the bones in 3 dimensions.
Here we use synchrotron light to study biological molecules. These biological molecules can be quite varied. We studied sugars, but also nucleic acids, such as DNA. But the most common biological molecule we study here on our beamlines are proteins. Proteins are very widespread molecules. They're present in all of our cells. They can be thought as the sort of molecular machinery that makes our cells function. They're also the main constituents of viruses. So understanding how these proteins function, how they assemble and interact with one another is essential in understanding how they function and how our cells function in our bodies. Obtaining this three-dimensional structure helps us to understand the functions of these proteins.
Here we are at the biomedical beamline. And synchrotron radiation is an extraordinary tool to visualize tissues and the inner part of the body. Our mission in imaging is to carry out development of techniques which one day hopefully can be applied also in hospitals. In the past years, we developed 2 techniques which are in preclinic able to treat much better and effectively cure the brain tumors. We have the possibility to target the tumor cells with a dose which is extremely increased. We can more efficiently treat the tumor cells with respect to the normal cells. This is an extremely important point in radiation therapy.
ID24 is a dispersive beamline dedicated to X-ray absorption spectroscopy. And we are dealing mainly with industrial partners, especially to investigate heterogeneous catalysts. A catalytic converter, what it does, it just transforms the 3 main pollutants, which are nitrogen oxides, carbon monoxide, and amber hydrocarbons, into the benign ones, which is nitrogen, water, and CO2. Synchrotron radiation plays a very important role because it allows to study these chemicals to an atomic level. So basically, we place the catalyst in a cell and we flow gases. So you can see dynamically what's happening at each time, how it is changing, which chemical reaction occurs, etc., etc.
Biomimetism is another very active field of research here at ESRF. Imagine if we could unlock the mysteries of spiders' webs. We could then copy Mother Nature to make a new material just as strong, elastic and light, the perfect rock climbing rope. Since ESRF opened in 1994, science has advanced by leaps and bounds. ESRF develops new instruments that help researchers push the frontiers of science even further. If history is any indication, tomorrow we will likely be conducting experiments that we can't even imagine today.